Bring a snack with you.
Bring a water bottle, a snack, leave it in the break room.
Bring a jacket. It's freaking cold. We tried to like not bring a jacket because we didn't want to store it the whole shift that we were there. Bring a jacket. So there's a place to put it.
I feel like it got really intense like the second half of the semester.
Wanna think holder? Sorry.
Oh, there we go. Great.
I have a lot of stories.
Okay, so hi everyone. I'm Victor. Just kind of looking around and seeing all the new faces. Okay, so did the video work okay for everyone for last week's session? Okay, cool. So I'm gonna record my sessions from now on, post them to the same channel or playlist or whatever. Also, right now I'm going to circulate my cheat sheet that I use for class. Yeah, thanks. Great, thanks. All right, so you guys can take a look at it and then just kind of pass it around and, whatever. So today's agenda, these are a couple things that people brought up last year as well as today for this week that I thought might be good to focus on. So Nernst equation, epigenetics, clinicals, acid base, innate immunity, how lab results might get formatted into questions, and we'll try to get as far into the immunity section as possible. And then next week we'll be returning to our normal 5 p.m. session downstairs in St. Mary's 104, and then we'll, since we started late, we'll be running a bit over time today. Thanks. Okay, so first off with the Nernst equation, or in general when you have equations in this class, I would suggest that you write them down on your cheat sheet, be familiar with all the variables that you see. So for example, with the Nernst equation, you have 1 over z, and that z is the charge, so you should know common charges like calcium +2, magnesium +2, sodium +1, potassium +1, chlorine -1. So all of those are basic ions that you should all be familiar with. Times 61 times the log of the concentration outside divided by the concentration inside. And so a practice question that he might give you is, a cell has an internal potassium concentration of that, external of that, what is it? And that's literally just plugging the numbers in, and just remember it's outside over inside. Okay, and I'll also send these slides to you guys after, so if you guys don't have to stress about writing every little thing down. So with epigenetics, this is just about once you have your DNA for the children, they have what's called methylation, or kind of these things that come after the DNA has actually been passed on. And so methylation, what it can do is it can silence certain genes, so certain genes don't get expressed and things like that. So you get some clinical manifestations, like with Prader-Willi and Angelman syndrome. And so Prader-Willi is a deletion of the father's part of 15q, chromosome 15, the Q meaning the long arm. And so with this, it's some forms of loss of coordination, and also just kind of physical features are different. And this is a very different disease than with Angelman syndrome with the mother's 15q portion deleted. And so this can lead to a neurogenetic disorder with developmental disabilities. So basically with epigenetics, it's good to know that concept, and if he asks you like deletion of 15q, father versus mother, one is Prader-Willi, one is Angelman's, it might just be a simple question on the exam. With clinical cases in general, I would suggest you guys pair up into groups of like 4 or 5 people, try to divvy them up and form like a study group with each other, and really like try to go over the cases together, or everyone divide them up and really get into the nitty-gritty of like reading the whole case and knowing what the whole case is about, and you explain it to other people. And when you do that, what you should do is you should like highlight only very significant portions. Like I would say like 23-year-old male, so like general age and sex of the person typically are the same, like almost word for word. And then you would look at lab diagnoses, like what things were elevated, what things were the same, if any tests were performed, or any bacteria that they found. And then also like very obvious things like this was positive for syphilis or something like that. And so this entire blurb, I would translate it down to, it's a case of HIV, 23M for 23-year-old male, diarrhea greater than 12 days, 12 BM for bowel movements, vomiting, anorexia, increased creatinine, increased sodium, decreased potassium, etc., etc. So I would just boil the whole thing down into a one-liner, and if you see on my cheat sheet, then like they're little like one-liners, and then you guys as a group can really like explain the whole case to each other, and that it's a more efficient way of getting through all of them. Because you guys are like a very tight-knit, group of like 20 students or whatever. So with here, similar thing. So like big blurb, and you can just boil it down to increase this, increase this, increase that. And that can really just help you in the long run. I guess we'll skip this one. Okay, so with fluid movement in general, you've got two types of pressure. You've got either hydrostatic, which is pushing, or oncotic, which is pulling. So inside like the arterial, for example, towards the arterial end, you've got capillary hydrostatic, so it pushes fluid out. But you've also got oncotic pressure pulling fluid back in towards the venous end. And in general, with osmosis, fluid goes like from high to low concentration. So in terms of solutes being like sodium, potassium, other ions, and other things floating around, those can kind of influence how much water you have. So for example, if I had more sodium, that would mean proportionally I have less water, so then I kind of go to where that area is. So that's the whole idea of osmosis. This is just very similar to what I was just saying. So depending on where there's more fluid, it'll always flow in the area of lower concentration. And so that could be from extracellular fluid into the cell, or it could be from the cell outward, depending on what their environment is. I think I put this chart on my cheat sheet. I think it's a good thing to know in terms of, or try to understand. So go through charts like these in general, and you want to look at, if this decreased, what happens next? If that decreased, what happens next? And you have to run through it yourself. So if I lose plasma proteins, for example, I'm decreasing capillary oncotic pressure. I'm not pulling as much. So it's like the example that I just gave with the sodium. So if I had more sodium, then I had less water. Likewise, if I have less proteins, then less, pull as well. So in general, I would run through these kind of figures. And so this figure is about ADH secretion. So ADH is anti-diuretic hormone. And so what you use it for is to kind of keep fluid inside the system. So if you are feeling that you have, that you need to, so if you have, are thirsty, for example, or if you don't have much water in the system, you increase thirst to build up your fluid intake. Or you would you would increase the ADH secretion so that you retain your water. And when you retain water, you're circulating fluid in your system more readily. And if you're circulating fluid, you dilute that plasma osmolarity back down to normal. And then because you're back to normal, you don't need the ADH secretion in general anymore. So this is a common example of what's called a feedback loop. So feedback loops are all about, if this happens, how do I compensate? Once I compensate, I should turn off the original signal to compensate because I don't need it anymore. Does that make sense? So one more time with this. If you have a low plasma osmolarity, if you need to increase it, then you would either drink more, or you would try to retain water with ADH secretion, and that builds up how much fluid you have overall. And if you build up how much fluid you have in your system, you dilute out the osmolarity, so plasma osmolarity decreases, and then you don't have to secrete ADH, and you don't have to have the thirst as much anymore. So it feedbacks to the original like signal for you to have the thirst and the ADH secretion, and then you can move on. So this definitely relates to the renin, angiotensin, aldosterone system. So with the ROS system, what you have is, if you have starting with low blood pressure, low blood pressure, you want to secrete renin from the kidneys. Once you secrete renin from the kidneys, it'll activate what's called angiotensinogen, and angiotensinogen will be converted into angiotensin 1, which will be converted to angiotensin 2 in the lungs by an enzyme called angiotensin converting enzyme, ACE. So once it becomes Ang2, you go back and you stimulate the adrenal gland, specifically the adrenal cortex, which is on top of the kidney, and then you stimulate the cortex to release aldosterone. So aldosterone is a hormone that'll make you retain sodium. And so, like I was saying a couple slides ago, when you have more sodium in the system, water will follow that high concentration of sodium to kind of dilute it out, because you had less water in proportion, so you want to go to that area where there's more sodium, meaning less water, and so you're keeping more fluid in the system. You're retaining water when you retain that sodium, and so it feeds back until you don't have to keep secreting the renin anymore. Does anyone have questions about this? Because this is a pretty important topic.
That would be an increase in blood pressure.
To increase blood pressure, we activate this system.
And once it's increased, that's when
Then it'll go away. Yeah. So that's kind of the opposite of this one here, which is the atrial natriuretic peptide, or hormone system. So ANH, or ANP, however you want to call it, its job is, if you have a high blood pressure, it wants to decrease your blood pressure to normal. So you secrete this peptide hormone, you release it, so you had a lot of sodium to start off with, and you want to get it back down to normal, so you release ANH, and what that does is it'll first decrease the ROS system, which would increase your blood pressure, and you don't want that. But also, you want to, let's see. So with ANH, you also want to get rid of the excess sodium in the system to make the water kind of go out with it. Does that make sense? So that's the opposite of the RAS system. So if you have high blood pressure, you can have the atrial natriuretic peptide system activate to get rid of the blood, to lower the blood pressure to normal. Okay. This is a chart for membrane excitability, and so basically there's two ions that can affect how easy it is for the cell to get activated. So in a normal case, you have the resting membrane potential, and that's determined by your potassium. And so potassium is the K. The K is what's determining what your cell is normally at. And so if you guys remember, for cell depolarization and stuff, it's the potassium that sets your baseline. Okay, so I have a baseline of negative 90 millivolts at this case. But if I had less potassium, I'd be less positive charge, right? So then I'd be even more negative. So I'd be even farther away from the threshold that I'm trying to hit. If I had more potassium, that's more positive charge, so I'm even closer to the threshold, so I'm more easily excited, okay? This differs from calcium because calcium is setting the actual threshold itself. So if I have more calcium, I would increase the threshold, so it'd be a farther distance to go to, versus if I had low calcium, I decrease the threshold, so I'm actually closer or more easily able to hit the exciting threshold. So basically it's the high potassium or low calcium that can make you more easily excited versus low potassium and high calcium that would make it more difficult to get excited. With this slide, it's just showing you the different components of an EKG. I wouldn't worry too much about this in this case. It's just to emphasize again that potassium and calcium concentrations will affect the electrophysiology of the system by making it more or less easily excited. Okay. You'll talk more about EKGs in a future lecture for more Unit 2. This is a chart for parathyroid hormone and calcium. And so calcium in the system is regulated by two of these main hormone secretions. So the first one is parathyroid hormone, and the second one is calcitonin. And so we're going to talk about parathyroid hormone in this case. So if I have too much calcium, I want to use parathyroid hormone to I don't want to use parathyroid hormone because parathyroid hormone, it releases calcium by one, it stimulates the intestines to kind of absorb more calcium back, and it also will break down some of your bones to release the calcium into the bloodstream. So if I have too much calcium, think of a feedback loop. So too much calcium, I don't want to release any more into the system, so I'm going to decrease parathyroid hormone, which is the hormone that would release more. So I decrease parathyroid hormone because I don't need any more calcium, and that would also lead to decreased activation of vitamin D and less absorption and other things like that, where you would get that calcium from. But on the other side of it, hypocalcemia, so hypo meaning too little or less, so low calcium, I want to stimulate parathyroid hormone to release into the system to activate vitamin D and to absorb it in the intestines, to do more absorption in the kidneys, and also to break down bone to release the calcium into the system. So there's a lot of different ways that you can get calcium into the system. So it's the intestines can absorb the calcium, the kidneys can absorb it. When the kidneys absorb calcium, they also excrete phosphate as kind of like the side effect. And then you can also break down bone to actually get the calcium. The bone is more like the last resort, and it's mostly like the kidneys or the intestine absorbing it, but this is a very important idea moving forward, that too much calcium in the system or too little calcium in the system affecting parathyroid hormone.
Yes. So phosphate, is it just like you want, when you have more calcium, you want less?
It's like phosphate is just kind of like in exchange for the calcium. It's like it does the opposite of like what you would need with the calcium. It's not very important otherwise. Yeah. So acid base, let's see. So with acid base, the way I think about it is, first let's start with the pH scale. So the pH scale, it runs from like 0 to 14. It technically can go above or below that, but it's very difficult. It means it's very basic or very acidic. So pH is the negative log of H+, okay? That's very important. So that means that a high H+ concentration is a low pH, okay? And each step on the pH scale going from 1 to 2, 2 to 3, 3 to 4, that's an increase in 10 times the concentration of H+, okay? So in the body, we have a pH normal value of 7.4, okay? But if I go from 7.4 to 7.3 or 7.4 to 7.5, that's actually a pretty big difference, okay? Because if I went from 7 to 8, that's a 10 times difference. And, you know, you break it down, like each step is, you know, X, Y, Z. So you might see these numbers and you think like, oh, 7.4 going down to 7.3 might not actually be a lot, but it actually is. Okay, it's a very significant number, each step on the pH scale. Okay, so with carbon dioxide combined with water, it has a very special reaction and it becomes H2CO3, carbonic acid. And carbonic acid, once it forms, it'll automatically go into H plus and HCO3 minus. HCO3 minus we also call bicarbonate, okay? And H plus you can also call a proton, or you can call it a hydrogen ion, just different names for it. So H plus is what determines pH, okay? Everything else is somehow related to the H plus concentration, which then relates to pH, but it's all about the H plus concentration, okay? So there's different ways you can kind of fix acid-base problems. One is with the lungs, respiratory, or two is with the kidneys, and that's metabolic, okay? So with our CO2, we have a normal value of 40 millimeters of mercury, and that can range from 35 to 45, roughly normal, and then bicarbonate is usually 24 millimolar, and that can range from 20 to 28 as well. But in general, when you see like normal lab values, like I would keep them on my cheat sheet. Those kind of things are very important to know. And also when you see them in clinical cases, you can kind of tell like, is this high or low? So let's move on with this. So in the lungs, if you breathe more at a higher rate, then you're expelling CO2. So every breath you intake is oxygen, but every breath you exhale is the CO2, right? So if I'm breathing faster, I'm getting rid of CO2 in the system. So CO2 relates to the H plus concentration. So if I breathed out faster, I would be losing the H plus because I'd be losing CO2. Does that make sense? And if I lose H plus, my pH goes up. Okay, my pH goes up when my H plus concentration goes down. And in the kidneys, the thing that you could do is you could either increase the rate of H plus secretion, you could absorb more bicarbonate, but I usually think of it in terms of H plus secretion. Okay, that's a more intuitive way I think about it. But you know, this complicated slide is basically just saying like, oh, you could reabsorb bicarbonate, oh, you could get rid of acid, something like that. But generally, it's that main idea. So it's either respiratory, like to fix the pH, or it's metabolic, kidneys. So looking at this diagram, I think it's a very important diagram, and you should study this one because there's usually 2 to 3 or 4 questions on the exam. And 2 will be like a graph, just like this. So when I approach the graph, I will ask myself, A, am I acidic or basic? B, is it respiratory or metabolic? And C, like, am I compensating for anything, or is there another factor that will bump it one way or the other? So let's start at the beginning. We've got 2 respiratory corners because the graph, the way it's read, is you have these like lines going downward that are all CO2 lines, okay? So the normal CO2 line is along 40, and that's why your metabolic problems are in the middle along this line here, because it's along normal CO2. And if you have more CO2, that means respiratory acidosis, because more CO2 is more H plus, and more H plus is a lower pH, so you became acidic. Okay, so the lower towards 0, like the lower your pH, the more acidic. The higher the pH, the more alkalytic or basic are the words, so that's why alkalosis. So respiratory alkalosis. I breathed out all my carbon dioxide, so I have less H plus in the system, which means my pH went up, okay? Then we look at the metabolics of it. So now we're looking at, okay, well, my CO2 is normal, so it's just a matter of like, am I acidic? Did I decrease my rate of H plus secretion? Like, am I holding on to acid in the system, or am I alkalytic? So am I excreting the H plus from the system? Okay, so that's how you break it down into these like 4 separate quadrants. And if you ever forget, just I would look at the CO2 to be like, okay, am I normal? Am I respiratory, or am I metabolic? I would look at the pH. Am I acidic, lower than 7.4, or am I basic, greater than 7.4? And then from there, I would think, okay, is there compensation involved, etc. So now let's talk a little bit about compensation. So with compensation, if I have a metabolic,
What's like a sample question, like with that previous?
it's gonna be like some sort of disease or condition, and it's gonna say he's gonna label like, you know, A, B, C, D, E, and he's gonna say pick one. Which one is it? So I think we have a sample question here coming up. So with compensation, if it's a metabolic problem, I can't fix it metabolically, can I? So I would fix it with a respiratory compensation. So I would use my lungs if I were acidotic. So if I have too much acid in the system, how do I get rid of acid? How do I get rid of H+? By breathing out CO2. Does that make sense? And if I had a respiratory problem, like if I was hypoventilating, if I wasn't breathing very much, I'd also be acidotic. So then I would have to have my kidneys increase secretion of the H+ to flush it out of my system. Okay? So that's how they kind of parallel. So one compensates for the other. Okay? So he provided this kind of chart of like how you could compensate and stuff like that. I would read it. If you want, you can always put it on your cheat sheet. In general, for me, I just try to remember what the different quadrants are, and I try to think again, like, how do I compensate it? One, respiratory or metabolic? Am I acidic or basic? And then overall, your end goal is to get as close to 7.4 as possible. And even if it doesn't look like you're compensating very much, like a 0.1 difference on the pH scale is a pretty big difference. Okay, always remember that. So maybe I took it out. Okay, well, sorry.
Can I ask a question?
Yes.
He said that we were gonna have to calculate base excess, but he didn't really put it on any of his.
Are you sure he said to calculate it?
Yeah, I asked him if we needed to know how to do that for the
Okay.
test, and he said yes.
But he didn't put it anywhere.
Okay.
I mean, I don't know if there's like a simple equation. We found like a crazy. I guess there's not a crazy one online.
I think it's on my cheat sheet.
Okay.
Yeah. Okay, sorry about that.
I don't know.
Yeah. So let me see. Base excess is 0.93 times, in parentheses, the bicarbonate concentration minus 24.4, end parentheses, plus 13.8 times, in parentheses, the pH minus 7.4.
Can you say that
I
again?
think that was on one of the slides, wasn't it?
I thought it was.
Was it?
Okay, so one more time, 0.93 times, in parentheses, bicarbonate concentration minus 24.4, end parentheses, plus 13.8 times, in parentheses, pH minus 7.4. And you guys can keep circulating my cheat sheet if you want. So it's in the bottom left corner. So going back to respiratory acidosis, common examples you'll see, because it's a respiratory problem, would be COPD or another acute respiratory problem. Because it is a problem with breathing, you can't breathe out as easily. If you can't breathe out as easily, you can't get rid of the CO2. And if you can't get rid of CO2, you build up H+, and you decrease the pH. Okay? Metabolic acidosis could be kidney failure, so that's renal failure, diabetic ketoacidosis, or long-standing diarrhea. So those kind of things, like your something is wrong with how much H+ is in the system because of your kidneys. Yes. Okay. Metabolic alkalosis it's seen with excess vomiting because you vomit out like all the acid, stuff like that. And then lastly, respiratory alkalosis. If you're breathing out too much, then you lose the H+ that way. So here are two samples. So it would just be the graph, and he would give you which one most corresponds. So the first one, let's think about prolonged vomiting. So if I'm vomiting too much, is that affecting? Is that a respiratory problem or metabolic? Metabolic, right. So if I vomit too much, I'm getting rid of acid. So if I got rid of acid, I would be alkalotic. So then I'd be on from 7.4 up. So that's either A or E. And then because it's a metabolic problem, I would say, is it A, which is close to 40, or E, which is close to 20 for the CO2? So I have normal CO2, so I would be A, close to 40. Going with chronic COPD, so COPD is a breathing problem, so if I can't breathe as easily, it's respiratory acidosis, right? So then from there, I look, okay, well, I'm on my acid side, so B, C, and D, which one has a CO2 problem? Which one has elevated CO2? It's only letter B, which is up here. So then that would be it. Does that make sense? So for these kinds of questions, it's always good to reorient yourself, narrow it down to a couple of choices, and then think from there. Yes?
What about, isn't chronic though normal pH
So
usually?
chronic can have compensation. So if it's for a metabolic problem, for example, you could compensate it a little bit with a respiratory. So you might sometimes see some sort of respiratory is off, but you see the pH is closer to normal.
Right.
So I would be careful with the word chronic. If it's a very blatant example like chronic COPD, then go with the more obvious answer. But if it's a chronic metabolic problem, that might point you in the direction of some sort of compensation, so some sort of respiratory compensation. Okay, so just read the question carefully, but don't overthink it, and don't underthink it. Easier said than done, right? So the Chem 7 basic panel, just throw it on your cheat sheet. I would, I guess on my cheat sheet, if you guys are looking there, the way I put it was okay, if I had increased sodium, what does that mean? I might be dehydrated, or if I had too little sodium, what does that mean? If I had too much BUN, that might be a kidney failure, or creatinine, kidney failure. And then I just kind of went through all of them and wrote down what they might all symbolize. And so you can look at my cheat sheet for what exactly I put for all of these values, and I can send that to you later
So would
as
you
well.
put another column? Or something?
If you look at my cheat sheet, I just squeezed it in there.
Oh, okay.
Yeah. Yes.
Would you recommend having all these charts on a cheat sheet?
Yes. Absolutely.
So everything.
Yes. Yes. So what he will do is he will I don't know if I have it. Yes, just like that. He puts all the numbers up there, and he says which of the following is most likely damaged or improper, and then it is your job to discern which one it is. We're not going to go through that right now, but you can always look for your own benefit. Same thing with these numbers. Be familiar with all of them. Yes. Okay. Going on to immunity, I don't think this was supposed to be the next slide, but sorry about that. It keeps skipping all the way to the end. Okay. Okay, so starting with bacteria. First off, does anyone have any questions about the lab medicine portion that we just talked about? Okay. All right, good. So with bacteria, I would just throw this chart on my cheat sheet, but beware that he did flip this in the original figure. So when you copy the figure, it does not copy the individual squares that change the top one to cocci and the bottom one to bacilli. So that is very important to note.
All the names on
Yeah, so I mean like
So he just put like a little box
so
for that.
yeah, so he originally had the word bacillus and cocci here, but it's actually cocci is at the top. You can tell from, staph and streptococcus. But then yeah, if you copy this figure directly, just make sure to change it on your actual cheat sheet yourself. Let's see. Going through the bacteria that you should know, Clostridium perfringens was involved with gas gangrene. If you guys might remember back a little bit, staph is one of those most common nosocomial infections, so hospital-acquired. Streptococcus can lead to strep throat, rheumatic fever. Let's see, listeria with Listeria monocytogenes. Like, I don't think you guys talk about that too much. And then let's see, Neisseria was with gonorrhea. Yeah, I guess we'll if we come up with these
He does the Clostridium a lot with C. diff.
yeah, so Clostridium C. diff or Clostridium perfringens and staph are probably the most common ones that you'll see, and then you may just see the names of other ones come up, but they're not going to be as difficult. They're not going to be like what is
Exactly
Bordetella.
what I did.
So, but we should know
But, like,
what the other ones are.
Yeah, like so the more common
told us
ones.
to kind of focus more on gram positive.
Yeah, gram positive, gram negative. That kind of stuff is very simple. I think I just mentioned them because like with Clostridium in specific, it did come up in a different PowerPoint as well. So if he asked you like which gangrene came up because of C. perfringens and that kind of thing. But otherwise, yeah, definitely is it a coccus, bacilli, gram positive, gram negative. So do you guys all understand what the difference between gram positive and negative are? Okay, cool. So it's basically, it has to do with the outer layer, this peptidoglycan layer, and so gram negative, it doesn't stain, while the gram positives do because of the way their outer layer is. DNA and plasmids, so bacteria, unlike humans, they will just reproduce in number, like asexually, and then the way they have the sexual transfer of information is through a process called conjugation, and so they literally will attach what's called a sex pilus to another bacteria, and they insert genetic material into the other one, and that's how they have the recombination. Viruses are non-living organisms, and they are basically floating around DNA or RNA wrapped up in some are wrapped up in a protein kind of envelope or capsid. And so with those, what viruses typically will basically do is they attach to your cell, and they make their way inside your cell, and then they open up, and then their genetic material kind of wiggles its way into your genetic material, and then it can either make more of its own self and make more enzymes to help facilitate that, or it can just embed itself in your genetic material and just kind of wait until your DNA replicates and stuff like that. So viruses in general are a bit more tricky because they're not actually living, so it's not like you can kill it or anything, but the whole point of a virus is always just replicating somehow, and it's trying to be as most advantageous as possible. Going to fungi, there's either molds or yeasts. So yeasts are single-celled, whereas molds are filamentous, and they reproduce differently, and the ones that you should know for fungi are with the Candida albicans, and that's your normal gut flora. And so gut flora, basically, it takes up space in your intestines so that other bacteria can't live there. So ones that are more harmful. Aspergillus with the respiratory infections, and then Tinea, capitis, curris, and pedis. So athlete's foot is one of the more common ones that you'll hear of. Yes. Now going on to the innate immune system. Okay. So I guess I would just run through this and make sure you know all of the different cells. What is the order? So never let monkeys eat bananas. So like neutrophils are the most, followed by lymphocytes, monocytes, eosinophils, basophils. And this chart can just kind of orient yourself. Like if you look at it, you should be able to identify like what it is on each of these. And we'll go more in detail. So overview of the immune system. So with humoral immunity versus cell-mediated. Okay, so humoral, like if you think back to the olden days of medicine, they didn't actually know that we had like blood circulating around, or they didn't characterize it as such. They characterized like 4 main fluids called humors. And so those humors would be like your blood, your urine, your, whatever. And that's why they thought that there was some sort of like magical property in your, in your humors. So that's what the B cells and myeloid cells kind of encapsulate. So they're cells that are constantly floating around in your bloodstream, just kind of looking for stuff that they can attach to and kill or deal with. So you have an innate system and an adaptive system. So humoral innate would be myeloid cells, and we'll talk more in a second. And so those cells just kind of, they see something in the bloodstream and then they'll kill it. Cell-mediated, however, is intracellular. So they have to find something, like something infected your cell itself. So it has to find that problem in the cell. So like a natural killer cell, it'll see a virus-infected cell and it'll kill the cell because it has a virus inside of it. So intracellular, okay? Whereas extracellular would be like some sort of bacteria or debris that's floating around in the bloodstream. B cells are more adaptive. They're very, very specific, and they only bind to things that they have a receptor for. So a common theme in all of biology is specificity. So one B cell will only bind to one type of antigen. Antigen is what the antibody binds to. And then a T cell is a little bit more complex, but it also is cell-mediated. So it finds a cell and it finds a problem intracellularly, as opposed to it doesn't go looking around the bloodstream for something. Okay, and we'll go more into that as well. So cells of the innate immunity. So in the innate immunity, we've got a bunch of different types of cells. So we've got like the neutrophils, the eosinophils, the basophils, mast cells, dendritic cells, macrophages. Macrophages are probably one of your biggest key players, because what they can do almost everything, like from engulfing the cell, and that's phagocytosis, so cell eating, versus they can secrete inflammatory cytokines, or basically they clean up debris, they clean up bacteria that come across their way. They do a lot of different things. So inflammatory cytokines will prime your body to be in a state of like, oh, something has infected us. Like, we should allow like the bloodstream to flow a little bit more, and allow like our macrophages to get to the site of infection more easily. So like there's a lot of things that go into this kind of immunology aspect of the body's inflammatory system, and we'll kind of see them as we go. So let's see, with this cell, with this picture, let's pick another one. So with this picture, what it's showing you is with neutrophils, they kind of prime themselves, and they want to like get to the position of like, okay, let's say in this tissue there's some sort of inflammation or some sort of invader. Neutrophils will come, and your body like will have vascular permeability, so it'll kind of spread apart a little bit more easily. Like blood flow will increase to get the neutrophils to that state, and then they'll be able to kind of go through and deal with the problem more easily. So like your body will kind of space apart just a little bit so that these kind of things all squeeze through. I don't think I'm explaining this picture as what there's a lot of things going on in this one, so we'll come back to it, or we'll probably deal with it in different ways, because I think this is a bit confusing. Acute inflammatory response. So you can have vasodilation, so more blood, because like if the blood vessel itself opens up, then you got more blood getting to the actual location, right? Vascular permeability, so like the cells are a little bit more easy to pass through. Cellular infiltration, that's pus, so that's the neutrophils actually getting through the more permeable area. Thrombosis is clotting, so a thrombocyte is another word for a platelet, and platelets are for clotting. And then you can also stimulate nerve endings, so that's pain. Mast cells are another part of that innate immune system. So what mast cells can do is they have this thing called degranulation, and so they've got a whole bunch of contents inside, and then you find usually it's some sort of parasitic worm, for example. You go up to that and you degranulate, so you spill out all of these factors. And so those factors could either be like histamine, which is for, it affects like your blood, or it could be chemotactic factors. So chemotactic factors are things that attract either neutrophils or eosinophils to deal with the problem. So other forms of white blood cells to deal with the problem. And then I guess this other side of the picture isn't as important, but I would still look at it. It's just synthesis of different things. Yeah, I would more focus on this idea of degranulation. Nature of leukocytes and inflammatory response. So, let's see. So if you have edema, so edema is buildup of fluid in the tissue, then the neutrophils will eventually, get to that spot, and then after they come, like macrophages will also come to kind of clean up the system and get rid of all this dead neutrophils, debris. So process of phagocytosis. So in phagocytosis, you have, sorry, I had a different slide that kind of condensed all of this. But in, let's break it down. So you have what's called pavementing, which is when the neutrophils will kind of all get up close to the tissue that you're trying to like fix, or then the cells will have increased permeability, vascular permeability, and they actually will open up a little bit, and then you allow for diapedesis, so the neutrophils can actually get into the cell, and once the neutrophils actually come into the cells, they go to these chemotactic factors. It's chemotaxis, go to the area where it has most of these factors and deal with the problem. Okay, so that's phagocytosis neutrophils dealing with the problem. So pavementing, getting up close, yeah.
Would some of the other cells deal with it the same way? Like
Yeah, so-
diabetes,
so
is this just like an example?
I think it's just an example. I
Okay.
mean, macrophages will also like
Do that.
come into the
Okay.
system and like do phagocytosis and other things as well. So in general, phagocytosis, you have to somehow get around the cell and then you just engulf it. Yeah, that's the idea of phagocytosis. With these cytokines, I would just know that there are these like different things that the macrophages can do. Like they can initiate a response, they can recruit other cells with inflammatory cytokines, they can remove debris by like engulfing it, and they can also promote repair and regeneration. Cytokines can, increase like fibroblast growth or collagen production just to fix the wound. So it's just kind of different things that get, the help to clear out the infectious agent and then to stimulate the growth to fix the problem.
So he said to know the stages and know like those blue boxes. Would you say we don't need another note?
I think I put it on my cheat sheet somewhere just to have.
Because I remember he said know the stages and I have them.
I- If you look here, like I have a very small, like couple word summary of all of these. The idea is pretty straightforward. It's like you have a problem, stuff is gonna come help you, and then you're gonna fix the problem. But it, in fancier words, yeah. This is pretty much the same thing that we've been talking about. So you can fix a problem, resolution. You can have inflammation chronically, so you can't fix the problem, but you keep trying to fix it. Or fibrosis, so your cells eventually you start to lose function, and everything is just very difficult from there on. But yeah. So the slides that I kind of glance over, they're ones that okay. Lymphocytes, adaptive immunity. So with lymphocytes and adaptive immunity, remember B lymphocytes are different than T lymphocytes, so B cells versus T cells. B cells come from bone marrow, T cells come from the thymus, and natural killer cells also come from the bone marrow. So pretty much all, except for the T cells, are bone marrow, and then T cells, thymus. And I move that over. Okay. Alright, so with your thymus, your T cells, what you have are cells that they are immunocompetent, and what you want to do is you want to select cells that will have a receptor. So these cells, they can function, they don't bind to your own cells because of some sort of selection process that they go through.
With the T cells.
Both B and T cells. They have some form of selection before they're released out into the world, okay? It's like priming, yeah. So with the T cells, they can only bind to things that have what's called an antigen-presenting cell, APC. And so the antigen-presenting cell could be a macrophage, could be a dendritic cell, could be a couple other things, okay? But basically what it does is it's like, here, I am a T cell, have this, antigen that I found for you. And then the T cell will be like, oh, I can bind to this. And then it may or may not kill the whatever it bound to, depending on what the process is. A B cell, on the other hand, does not need the antigen presenting cell. It can just float around. If it sees it, it binds to it, it'll inactivate it, or whatever it'll do, whatever the function. So we have all of these different things like Peyer's patches, lymph nodes scattered throughout the body. And so when our cells are first released into the system, they want to find their antigen. And once they find their antigen, that's when they truly become activated. That's when the body knows, oh, you're pretty useful. You actually found something. So now we make more of you. Otherwise, you've got literally billions of different possibilities that are always floating around the system, kind of looking at the different parts of your body, looking for that antigen. If you don't find the antigen, you remain inactive. But once you get activated, the body's like, hey, we have use for you, and then it can circulate more. Let's see. So an antibody has this kind of generic Y shape. Let me see if there's a... Yes. So it has this Y shape, and then the bottom portion is called the FC domain. It's the constant part of an antibody. It's the same for all of them. But then you've got these heads that are part of the light chain, and they are the variable region, okay? So in 3 dimensions, this is what it looks like, but simplistically, it's it's a Y. So a macrophage, for example, or a T helper cell, or other things can bind to this constant region if it needed to. Like for example, if I had an antibody that I wanted to tag with something, it would bind to my antigen, and then I could find the antibody by the constant region. But yeah, that's what an antibody looks like. So there's a whole bunch of different Ig letters. So there's IgA, which is for the GI, bronchorespiratory, genital urinary tract. IgE is like mast cells secreted in tissues. It binds to mast cells for like parasitic worm infections or allergies and stuff like that. IgG is kind of the general antibody of the blood. IgM is like the kind of the precursor to everything, and then IgD we don't know what it does. And again, I'll be sending these slides later. So antibodies are highly specific. They only bind to what the light chain receptors can bind to. They can inactivate the cell in a couple different ways. So they can physically, like each arm can bind to some sort of antigen, and each arm is the exact same. They bind both to the same thing. Okay, so one entire antibody goes for one antigen, one type of antigen. They could bind to a bacteria by inactivating. So the bacteria is the little thing. The bigger, like Pac-Man looking thing is the macrophage. So remember how I said they all have the same constant domain? And so the macrophage has like a receptor for the constant domain. So it can bind to any antibody that has bound to a bacteria, let's say, or other kind of thing. They can also inactivate all the binding sites of a virus, for example, of a bacteria, of a toxin. They can, I think what's shown here is they kind of built up this platform for another cell to come in and like punch a hole into the bacteria, or they tagged a parasitic worm for a mast cell to come and degranulate over the parasitic worm. So there's a whole bunch of different types of ways that the antibody can help the cell. With B cell fundamentals, we've got a whole bunch of different types, and you have what's called a nurse cell come present to the initial, like, it presents a self-antigen to screen out for any possibilities that that antibody could attack us. So they do not become active until they pass that negative selection test. So you do not want it to bind to your self-antigen. And you can have billions and billions of different types of B cells, and it's got a higher error rate because you want error rate in making the DNA, because you want tons and tons of variability, because you never know what you could come across in the cell. So again, a nurse cell will present your self-antigen, and then your body will say, oh, you bound to a self-antigen. We're going to kill you before you go into the bloodstream and kill us. IgE-mediated destruction of a parasite. So again, with this example was, first we have the parasitic antigen, whatever, like scraped off from the outer layer or something like that. Then we stimulate a B cell. So a B cell is what, gets rid of or makes the antibodies. Like each receptor on a B cell is an antibody. BCR, B cell receptor. And so all these receptors, in this case, are IgEs. The IgE will bind to the parasitic antigen. The mast cell will bind to the antibodies. It will send some sort of chemotactic factors for the eosinophils to come, deal with the worm. So all of those kind of things relate to each other. And so this was like a past sample question, like rank all of these in order, and then we can just we can you all can run through this on your own afterwards, but it's that idea that like I think the answer is D in this case, or 4. So local APCs present worm antigen, the B cell activates, then you have plasma cells, which is the B cell, producing IgEs, then you went back, the antigen binds to the IgE, then you